Puccinia graminis, habit and habitat. P. graminis is an obligate parasite, polymorphic, macrocyclic and heteroecious rust. It affects wide range of hosts including wheat, barley, oats and rye. Grass hosts include Agrostis, Dactylus and Agrophyron. P. graminis tritici involves in its life cycle two distinct alternate host plants, i.e., wheat Triticum sativum, and barberry Berberis vulgaris. The wheat plant is called the primary host and the barberry plant is secondary or alternate host. Symptoms on wheat. The symptoms of the disease appear as large, elongated and brown pustules Uredospore on the stem, leaf, sheath and leaf. Later on these brown pustules change into black-colored large pustules Telatosauri. Grains of the infected plants are shriveled, much lighter in weight and thus reducing the yield. The pathogen shows a balanced host-parasitic relationship. Even in the severe infection, the parasite does not cause much serious damage except that the growth of the plants may be somewhat retarded and the granis may be of reduced size and of poor quality. Symptoms on barberry. Infection first starts on the dorsal surface of the leaf in the form of minute, dark-colored and flask-shaped Puccinia which appear as yellow spots. Beneath Puccinia, on the ventral surface, appear cup-like projections of Aecial or Acidia. Vegetative structure. The mycelium is dikaryotic. Each cell of the mycelium bears two nuclei, on primary host wheat, and monokaryotic. Each cell of the mycelium bears only one nucleus, on the secondary or alternate host barberry. The mycelium is well developed, branched, septate and does not spread throughout the host, but is localized to isolated patches. It is either intercellular or intracellular, with the former producing bulbous, branched or knotted haustoria into the cells for obtaining nourishment. The transverse septa are present or long intervals between the cells. Each septum contains a simple central pore. Cell wall is made up of fungal cellulose. The cytoplasmic membrane surrounds the granular cytoplasm and reserve food material remains in the form of glycogen bodies and oil globules. Life cycle. Puccinia graminis is long-cycled rust macrocyclic. At the time of reproduction it produces 5 distinct stages in a regular sequence. These are as follows. Stage 0, Spermagonia bearing spermatia and receptive hyphae. Stage 1, Aecia bearing Aeciospores. Stage 2, Uredia bearing Uredospores. Stage 3, Telial bearing Telatospores. Stage 4, Promycelia bearing Basidiospores. Out of these 5 stages, Uredo stage, Telato stage are produced on the primary host wheat, and remaining 2 stages, Spermagonial and Aecial stages, are produced on the secondary host, i.e., barberry. Stages on primary host wheat, Uredospores or Uredo stage. This stage is formed by the infection of the Aeciospores brought from the infected barberry plants or by the Uredospores themselves coming from the neighboring wheat plants infected earlier. Both the spores are binucleate and on germination, produce a germ tube on wheat leaf. The germ tube grows over the surface of the epidermis of the host and on reaching a stoma its tip swells up and forms a vesicle-like structure called oppressorium. The protoplasm of the germ tube migrates into the oppressorium. Now it is cut off from the germ tube by a septum. The oppressorium produces a narrow hypha. It enters inside the substomatal chamber through stoma. Its tip again swells up and forms a substomatal vesicle. The contents of the oppressorium migrate to vesicle through narrow hypha. A hypha of the dicarion 2 nuclei cells develops from this vesicle. It branches and produces hyphae which spread in between the cell, intercellular, but occasionally produce hausotria. Telatospores or Telial stage. Towards the end of the growing season of wheat crop, the environmental conditions become unfavorable, hot and dry, for the growth of the Uredospores. Now Uredosauri produce another kind of spores called Telatospores. First, they develop among the Uredospores within the same sorus, but later they develop in separate sori known as Telatosauri or Telutopustule. As the crop matures, the number of Uredospores is reduced and the sori contain only Telatospores. This stage is known as the black stage and hence the name black rust is given to the disease. The Telatospores are dark brown or black in color. They are bicelled and spindle-shaped structures with a pointed apex and thick smooth wall. Each cell of a telatospore has a single germ pore and 2 nuclei, one of plus strain and the other of minus strain. As the telatospores reach towards maturity, choreogamy takes place and the 2 nuclei fuse to form a diploid nucleus. The development of telatospores is entirely similar to the Uredospores. At this stage the telatospores undergo a period of rest. During resting period they lie on the ground or still attached to the host. These are the dormant cells and are capable of tiding over unfavorable period. Basidial stage, after the renting period, the telatospores germinate during the early part of spring. They germinate in situ and either one or both of its cells give rise to a germ tube, known as promycelium. The promycelium together with the telatospore cell is called basidium. However, many authors prefer to call the telatospore cell as the hypobasidium and the promycelium as the epibasidium. The diploid nucleus of the telatospore migrates into the promycelium and divides meiotically into 4 haploid nuclei. The septa appear between the nuclei and divide the promycelium into 4 haploid cells. Each haploid cell of the promycelium produces a slender, short, lateral, tube-like structure known as sterigma. The sterigma swells up at the end to form a spore-like cell. The haploid nucleus from each promycelium cell migrates into this developing spore cell through its respective sterigma. Thus, at the tip of each sterigma, a minute spore is formed. This spore is called basidiospore. Each cell of promycelium produces a single basidiospore. Thus, from a single cell of telatospore 4 haploid, unicellular, uninucleate basidiospores are formed. 2. Out of the 4 telatospore basidiospores are of plus strain and the other 2 of strain. Soon after the basidiospore formation they are forcibly ejected by the water droplet method. In this method a liquid begins to collect in the form of a droplet at the base of the basidiospore. This droplet gradually attains a bigger size and suddenly pushes off the basidiospore forcibly into the air to a short distance. The basidiospores are carried away by wind. They are capable of germinating only on barberry plants Berberis vulgaris available on hills. They perish soon if the alternate host is not available. Stages on barberry plant, the basidiospores, which fall over the upper surface of barberry leaf start germinating soon. They germinate by giving out a germ tube which penetrates through the epidermis. The germ tube elongates and divides inside and develops into hyphae. These hyphae grow between all the cells lying in between the lower and upper epidermis. The hyphae are composed of uninucleate haploid cells primary mycelium and are of plus or strain. Several basidiospores of different strains may infect the same berberis leaf. Thus, haplomycelium of 2 different strains plus or is formed. It remains haploid for some time and the fusion between these 2 strains plus or may occur at a later stage. Spermagonial or pycnidial stage. This stage is also known as Puccinia or spermaceal stage. After about 4 days of the infection, the haplomycelium collects and forms dense mats both beneath the upper and lower epidermis. The mycelial mats beneath the upper epidermis are known as primidium of Spermagonium while the mats beneath the lower epidermis are known as primidium of Aecidium or protoacidium. In 7 to 10 days after infection, each primidium of Spermagonia matures into a small flask-shaped structure called spermagonium or PVC nidium. The pycnidia appear as minute yellowish specks on the upper surface of the leaf. A vertical section through these specks reveals that each spermagonium opens on the upper surface of the host leaf through a pore-like structure known as ostiole. Its wall consists of 3 kinds of hyphae: sad face I, periphyses E, flexuous or receptive hyphae E, spermatiophores or pycnidiophores. Aecial or acidial stage, the haplomycelium forms the primidium of Aecidium or protoacidium beneath the lower epidermis. The further development of protoacidium into Aecidium takes place only after the dikaryotization. The spermaceal nucleus male nucleus by mitotic division forms a second male nucleus, which moves to the next cell, through septal perforation. In this way the male nuclei produced by successive mitotic divisions pass down and all the cells of primary mycelium are dikaryotized. The dikaryotic basal cells of the protoacidium arrange themselves vertically beneath the lower epidermis and are called as sporophores. Each binucleate basal cell then cuts off a chain of binucleate cells in basipetal succession on the side towards the lower epidermis of the host. These cells are the acidiospore mother cells. These cells further divide transversely to form a large cell and a small cell. The large cell develops into acidiospore while the small cell remains sterile and is known as disjunctor or intercalary cell. The latter dissolves and sets free the aeciospores. With the development of the aeciospores, some of the basal cells lying at the periphery of protoacidium mature into a one-celled thick protective layer called peridium. This entire structure is cup-shaped and is known as aecium. The developing Aeciospores rupture the peridium by exerting a pressure on it. Thus, the Aeciospores are liberated. They are unicellular, polyhedral, thin-walled, binucleate and orange-yellow colored. Germination of Aeciospores. The Aeciospores are disseminated by wind. They are capable of immediate germination but cannot infect barberry plants. Falling on suitable host, i.e., what leaf, they germinate by producing a germ tube or primary hyphae. The further development of the germ tube is similar as described in the uredenal stage, and ultimately the dikaryotic mycelium is produced. This is the mycelium which produces the uredospores and later the telatospores on wheat. In this way, the life cycle of Puccinia graminis is completed.